Bright screens and midnight snacks create a profound biological paradox. The pupils register stimulating, harsh light, while the gut lining processes a heavy bolus of glucose at a time the system expects to be dormant. The circadian clock governs metabolic and proliferative homeostasis, acting as the primary timekeeper for the body's internal chemistry. When light cues tell the brain it's midday, but food intake tells the gut it's midnight, the internal pacemakers fracture. The neurological clock fires at a completely different rhythm than the digestive organs below it. This state of temporal decoupling is known as chronodisruption. By forcing the body's subsystems to operate out of phase with one another, chronodisruption drives physical wear and tear, accelerating the canonical hallmarks of aging. The central pacemaker of this system resides in the SCN. The SCN, or suprachiasmatic nucleus, is a tiny region of the brain's hypothalamus that receives direct photic input from the retinas, entraining its rhythm strictly to external light-dark cycles. For decades, the SCN was thought to be the sole timekeeper. We now know that isolated autonomous oscillators, called peripheral clocks, exist in virtually every metabolically active cell, including those in the liver, skeletal muscle, and adipose tissue. To maintain order, the SCN acts as a neuroendocrine conductor. It attempts to synchronize these thousands of peripheral clocks by orchestrating the rhythmic secretion of systemic hormones, primarily cortisol in the morning and melatonin at night. There is a critical override built into this hierarchy. While the central SCN is driven by light, the peripheral clocks in your organs are primarily entrained by nutrient intake. When feeding times ignore the solar cycle, the body's metabolic organs physically decouple from the brain's neuroendocrine signals. The liver and muscles shift to a different time zone than the SCN, generating a toxic internal state of metabolic jet lag. To understand how a late meal overrides the brain's signals, we have to move from the macroscopic organ level down into the nucleus of a single peripheral cell. Here, time is tracked by a precise mechanical engine. This diagram illustrates the core molecular clock, starting on the right with the positive arm of the transcriptional feedback loop. The proteins clock and BMAL1 bind to the E-box promoter sequence, activating the transcription of the period, or PER, and cryptochrome, or CRY, genes. This initiates the negative feedback phase. Over the course of the day, the newly synthesized PER and CRY proteins accumulate in the cytoplasm, physically pairing up to form a repressor complex. To complete the cycle, this complex requires a chemical trigger. The enzymes casein kinase 1 epsilon and delta add phosphate groups to the proteins, allowing the PER-CRY complex to cross back into the nucleus and shut down its own creation by inhibiting clock and BMAL1. An auxiliary loop runs in parallel to stabilize this 24-hour rhythm. Down here, the nuclear receptors REV-ERBA and OOR compete for the exact same binding site to alternatingly repress and activate the transcription of the BMAL1 gene itself. This continuous cycle of transcription, translation, and precisely timed destruction by ubiquitin ligase enzymes serves as the ticking mechanism of every cell in the human body. Ubiquitin ligases are specialized enzymes that tag specific proteins for degradation by the cell's waste disposal system. The timing of this molecular loop is not isolated from what you eat. Nutrient sensors are physical gears plugged directly into the core circadian oscillator. Consider SIRT1, an NAD-dependent deacetylase that acts as an emoji sensor in the cell. SIRT1 physically binds to the clock protein and removes acetyl groups from BMAL1 and PER2, altering their molecular function. AMPK acts as a crucial checkpoint. AMPK is an AMP-activated protein kinase that senses fasting and cellular energy depletion. When energy is low, AMPK phosphorylates and destabilizes the CRY1 repressor protein while simultaneously regulating SIRT1. Consuming calories after dark forces a spike in insulin and mTOR. mTOR is the mechanistic target of rapamycin, a primary protein complex that drives cellular growth and suppresses fasting signals. The resulting clinical phenotype is severe. The nutrient-driven mTOR spike directly conflicts with the SCN's nocturnal signals, inducing nocturnal insulin resistance, elevating systemic inflammation, and disrupting the rhythmic release of appetite hormones like leptin and ghrelin. Because these nutrient sensors physically interlock with the clock genes, diet acts as a mechanical wrench. Eating at the wrong time literally rewinds or breaks the cellular timer. This mechanical disruption extends beyond metabolic organs into the central nervous system. The brain requires a strict, synchronized temporal window to execute cellular cleanup. A synchronized dark and fasting phase naturally downregulates mTOR and upregulates transcription factors like CEBPB. CEBPB is a protein that binds to DNA to stimulate the expression of autophagy genes, initiating deep cellular repair. This process drives glymphatic clearance and the destruction of dysfunctional mitochondria that produce dangerous ROS. ROS, or reactive oxygen species, are highly reactive molecules that cause severe oxidative damage to cellular structures if left unchecked. Autophagic vacuoles systematically engulf and break down these damaged organelles. Eating around the clock maintains constant mTOR activation. This sustained anabolic state permanently closes the circadian autophagy window. Without this daily period of deep clearance, toxic proteins accumulate in neural tissues. In humans, this manifests as memory impairment and conditions like sundowning syndrome, where dementia patients exhibit severe agitation and confusion in the late afternoon and evening due to fractured circadian regulation. Neural degeneration is heavily exacerbated by the loss of this circadian autophagy window. Without its designated daily temporal fasting period, the brain cannot wash away its accumulated metabolic waste. The tension between growth and clearance highlights the central paradox of aging biology. We see directly opposing effects when manipulating the mTOR and IGF-1 signaling pathways. IGF-1, or insulin-like growth factor 1, is a hormone that manages the effects of growth hormone to promote tissue anabolism. The lifespan argument is clear. Suppressing mTOR and insulin signaling maximizes autophagy, minimizes oxidative stress, and significantly extends systemic longevity. The muscle argument counters this completely. Chronic suppression of mTOR simultaneously degrades skeletal muscle tissue, leading directly to sarcopenia. Sarcopenia is the age-related progressive loss of skeletal muscle mass and functional strength, a primary driver of frailty and mortality in the elderly. A robust circadian clock is the only physiological mechanism capable of resolving this paradox. It routes these mutually exclusive biological states into distinct, separated time zones. Healthy aging requires a high-amplitude clock that swings the biological seesaw fully in both directions. It provides intense, temporary anabolic windows driven by daylight and feeding to maintain muscle mass, strictly followed by deep, restorative catabolic windows at night for cellular cleanup. This chart demonstrates the ultimate metric of chronobiological health, amplitude. Aging is characterized by a mathematical flattening of biological rhythms, visible here as dampened sine waves. This loss of peak to trough distance translates directly to physical decline, blunted hormone secretion, fragmented sleep, and chronic insulin resistance. We can restore this lost amplitude through non-pharmacological interventions like caloric restriction and TRF. TRF, or time-restricted feeding, is a dietary pattern that confines daily caloric intake to a consistent, shortened window of usually 10 hours or less, extending the nightly fast. Restoring the circadian amplitude yields cascading metabolic benefits. It lowers mitochondrial membrane potential, decreases reactive oxygen species output, and protects nuclear DNA from oxidative damage. When the amplitude of the clock is preserved, the neuroendocrine hierarchy functions as intended. Hormones like melatonin can accurately convey the rhythm of the SCN to peripheral tissues, modulating glucose-stimulated insulin secretion precisely when the body needs it. Longevity depends on meticulously respecting the evolutionary temporal architecture of the organism, synchronizing metabolic intake with the internal ticking of the biological clock.